In today's video, we'll be describing and talking about the molecular characteristics of contractile filaments found within the functional unit of the skeletal muscle cell called the sarcomere. This right here is the sarcomere. I'm going to identify the individual components. This is myosin. That is actin, the green. This right here is called titin. And that centerpiece right in the middle is called nebulin. Okay. Basically, the whole idea with sarcomeres is that this myosin head is going to come up here and attach onto binding sites, which then will push and contract it forward, causing a cascade of movements and thus a contraction of a muscle. Looking at the individual components of this of the actin, we see G-actin coupled proteins highlighted here, just like that. Okay, and that's surrounded by tropomyosin. Tropomyosin blocks the binding sites on G-actin, and what I was explaining for myosin is going to come up and it's going to bind to these spots, but tropomyosin is actually blocking the active sites. Okay, and then we have troponin here. Troponin is basically what allows us to remove tropomyosin from the G-actin active sites. So tropomyosin is composed of three proteins. We can take a look at it here. We have tropomyosin T, I, and C. Okay, troposin T controls tropomyosin. Troponin I controls the G-actin and like the stability of it. Troponin C, that's where calcium binds, and it has 2 low and 2 high affinity binding sites. So, basically, just a super basic rundown. Calcium, whenever you think about calcium equals muscles. Okay? Calcium will bind to the troponin sites, which will have a conformational change, essentially removing the tropomyosin. Now we're going to take a deeper look into this. I feel like we've covered tropomyosin, right? It surrounds the G-actin. G-actin is a bunch of just proteins that create polymers, basically, and troponin is what allows us to remove tropomyosin. Okay, we're going to take a look at myosin. Okay, myosin is skeletal muscles, and myosin type 2 is composed of, I didn't write this out, it's composed of 4 light chains and 2 heavy chains. Okay, the basic idea with myosin is that the myosin heads are ATPase enzymes, so that means they break down ATP. This breakdown allows for them to ADP to bind to actin. Okay, when it's bound like that, it's in a cocked state. However, when we remove the phosphate, that leads to a power stroke, or the contraction of the whole muscle. Now, in order for us to reset this whole process, ATP then comes into play here and binds to the myosin head, which allows us to reset, and it becomes uncocked. Okay. Now we're going to run through the basically this is the entirety of, I guess, the contraction of a muscle. So we're first going to start off with a signal going down a neuron. When this signal happens, right, we have voltage-gated sodium channels, which will release sodium into the cell, right, allowing for that Wong to continue, correct? And once we reach this voltage-gated calcium channel, right, again, if you remember, we'll have that influx of calcium. It binds, calcium binds to these circle proteins, and once they're bound, and they also bind to the vesicles which contain acetylcholine, right? They bind here, and it pulls it down into the membrane and releases acetylcholine into the synaptic cleft. Okay, once acetylcholine is released in the synaptic cleft, it will bind to ligand channels, and this allows openings, allows them to open, and sodium will move in super fast because it has a super high driving force. Sodium moves in and depolarizes the cell, okay? And I guess basically what you need to know here, make sure we're still recording. Basically you need to know here is that it's actually occurring in this green space that I just wrote down. Okay, it's occurring there. And we'll take a deeper look into that. See this Wong, right? It's gonna come through. We depolarize the extracellular space. It's depolarizing. It's gonna run into a voltage-gated calcium channel called right here, ryanodine. Ryanodine then activates. It's gonna run in. It's gonna the action potential will hit this protein here, which is called dihydropyridine. Dihydropyridine is a voltage-gated calcium channel. This calcium channel will interact with ryanodine, which is connected to the sacroplasmic reticulum, or the SR, which is this ryanodine is mechanically a calcium channel. They are connected. So when dihydropyridine receives this action potential, it opens up ryanodine. We'll take a closer look at that. Because ryanodine opens up, calcium from the SR is able to flow outside. It flows outside of the SR, and essentially what you would see here is, this is really horrible drawing, but basically this is actin. You would have tryptomyosin. Calcium is going to flow out, just like depicted in the picture. It's going to flow out, and it's going to bind, right? It's going to bind to troponin C. Once it's bound to troponin C, tryptomyosin will release. I guess I should show that. It's down here too, but tryptomyosin will release. Myosin then bonds here, allowing for that reaction to happen, or the contraction to happen. But we also need to remember that if calcium stays put, then we can't go into a relaxed state. So calcium has to be taken up into the SR through circle pumps here. And that's just a rough outline of the whole process of muscle contraction and the molecular components of actin and myosin, and how muscle contractions work through muscle cells.